Good day, everyone. My name is Janika Maynard, and for our final case presentation for Medical Biochemistry Unit 4, we are going to discuss on case number 4, which is a presentation of megaloblastic anemia. So what is megaloblastic anemia, you may ask? This is the presence of giant red blood cells, precursors, called megaloblasts in bone marrow. This anemia may be caused by the impairment of DNA synthesis, which is the result in division of the nucleus being inhibited. So, what are the causes of megaloblastic anemia? Megaloblastic anemia is due to vitamin B12 deficiency, among others. Vitamin B12 is very essential for DNA synthesis. So, how does our body gain vitamin B12 as our body does not create its own? Vitamin B12 is first absorbed in the ileum after it is bound to the intrinsic factor made by the gastric peripheral cells in the duodenum and the jejunum. Pernicious anemia is the most common cause of vitamin B12 deficiency, and other conditions such as Zollinger-Ellison syndrome, blind loop syndrome, fish tapeworm manifestation, and pancreatic insufficiency can also impede upon vitamin B12 absorption. However, the body also uses iron as a critical mineral to form red blood cells, which is required for a strong and healthy immune system, proper brain function, muscle power, and vigor. Most of its contribution goes towards the creation of hemoglobin, which is a rich protein found in red blood cells. So anemia, this is a disorder in which the blood does not have enough healthy red blood cells circulating in the body. And an iron deficiency anemia is a prevalent type of anemia, as the body cannot make enough hemoglobin, which is essential for red blood cells to carry oxygen around to our various organs. But an interesting fact, vitamin B12 is necessary for the synthesis of red blood cells, and a deficiency of vitamin B12 is the most problematic cause of anemia to develop. So a deficiency of vitamin B6 can also result in the reduced synthesis of heme, whereas vitamin B12 deficiency may result in a much larger than usual blood cells linked to the presentation of megaloblastic anemia. So vitamin B12 deficiency may also impair purine and pyrimidine metabolism by lowering the availability of the coenzyme methylene tetrahydrofolate. The coenzyme methylene tetrahydrofolate is required for thymidylate synthase, methylene tetrahydrofolate, or as an actin dihydrofolate reductase inhibitor. To wrap up this case on megaloblastic anemia, there are a list of potential laboratory tests and findings that would aid in the differential of megaloblastic anemia. This includes conducting a complete blood count, doing a bone marrow smear, and peripheral blood smears, as well as a CBC, MAT, low hemoglobin, elevated mean corpuscular volumes. So this is a test that measures the average size of your red blood cells. Also, decreased white blood cells and platelets may help in this differential. Peripheral blood smears shows macrocytosis and hypersegmented neutrophils, whereas bone marrow examinations may show myeloid soils and megakaryocytes. So treatment of megaloblastic anemia may include the supplementation of vitamin B12 or folate, and also necessary dietary changes to increase the intake of vitamin D12. Now for our final case, which is case 1, we are going to be discussing about ammonia transfer in the body. So the process of transamination and deamination is responsible for the production of ammonia by cells through the metabolism of amino acids and other compounds that contain nitrogen. This process is catalyzed by the aminotransferases and transferases in the body, which results in the formation of non-essential amino acids from essential amino acids, such as glutamate, which forms free ammonia for the synthesis of urea. Glutamate also forms free ammonia for the synthesis of urea through the oxidative deamination process. Deaminases are enzymes that catalyze the deamination of a molecule, which results in the excretion of ammonia from the body. Moreover, urea is converted from ammonia by the glucose-aldane cycle in the liver, which is filtered by the kidneys and excreted in the form of urine. Lanine is formed by the transamination of pyruvate in the glucose-aldane cycle as well, which is delivered to the liver through aldane transaminase. Glutamine synthase is a non-toxic ammonia carrier that catalyzes the condensation of glutamate with ammonia to produce glutamine. Glutaminase is a mitochondrial enzyme that catalyzes the formation of glutamate by the deamination of glutamine as it is transferred to the liver. The free ammonia then enters the urea cycle, and the remnants of glutamate is transaminase to produce alpha-ketoglutarate. The mitochondrial enzyme glutamate dehydrogenase catalyzes the oxidative deamination of glutamate to alpha-ketoglutarate, or it may catalyze the reductive amination of alpha-ketoglutarate to form glutamate. So glutamate dehydrogenase plays an important role in the fixation of ammonia from or into glutamate and the release of ammonia, which is essential for hemostasis. Glutamate dehydrogenase also catalyzes a reaction that leads to the continuous supply of intermediates for the TCA cycle, such as anovulation. Glutamate dehydrogenase is allosterically regulated by the activation of ADP, which facilitates glutamate oxidation upon a reduction in the cellular energy status. Finally, nitrogen transport is connected to glucogenesis in the fastest state using amino acids when the carbohydrate sources are depleted in the body. The glucose-aldane cycle transports ammonia from muscles to the liver. A deamination reaction converts amino acids to ketoacids catalyzed by glutamate dehydrogenase, glutaminase, and or aminotransferase. Conclusively, ornithine transcarbamylase, also known as OTC, is the most likely deficient enzyme in this case, and carbamyl phosphatase synthase 1 is the enzyme least likely to be impaired. Research has shown that arginine is the amino acid that becomes most essential due to OTC deficiency, leading to a reduced production of citrulline, which will ultimately lead to the deficiency of urea cycle of the urea cycle and production of amino acids. Thank you for joining in for our final case presentation for Unit 4. It has been a pleasure being a part of this class. Thank you so much.